Hello everyone, how are you? Today we are going to pass a new topic. The theme is cultivation of plants. In nowadays, agriculture plays an important role in economic growth of the rural people. Plants are domesticated on the basis of their varied applications as per development of human civilization. Plant utility is categorized like use as for food, fodder, fuel, spices, timber, fiber, and medical purposes. Among a varied group of the plants, many are grown widely whereas some species are cultivated per year or one or more times in a year based on climatic conditions and demand on the area. So on this lesson we are going to know how plants grow and develop. And by objectives you will learn to compare seed germination in beans and corn, contrast annuals, biennials, and the perennials, explain how primary and secondary growths are produced. A seed contains a plant embryo. You know it from the previous lesson that sperm and egg makes an embryo, after zygote of course. That is in a state of suspended animation. We will show it later. A seed cannot sprout until water and oxygen penetrate the seed coat. If enough water and oxygen are available after the seed coat breaks, a young plant or seedling beginning to grow. Germination. A plant embryo resumes its growth in a process. The first sign of germination is the emergence of the embryo's root. After the shoot of seedling emerges, its roots and shoots continue to grow throughout its life. From this picture, you can see the seed germination, how bean and corn they are growing and cultivates the plant. On the first picture, you see the hooked shoot of germinating bean seed straighten after the cotyledon emerged from the soil. And from the second picture, you see the shoot of germination corn seed is covered by sheath. The shoot grows straight up, but the cotyledon stays underground.
When a monocot plant seed, like this corn kernel, is dropped in an environment with water, oxygen, light, and the correct temperature, it may germinate. Monocot seeds develop a single cotyledon, or seed leaf. The radicle is the embryonic root, and its appearance is the first sign of germination. The plumule is the embryonic stem and leaves. Endosperm is nutritive tissue that supports the growth of the embryonic plant. The corn plant has a protective sheath that grows around the shoots. The cotyledon remains underground. Eventually, the first leaf emerges from the protective sheath. The plant is now a seedling. The roots and shoots continue to grow throughout its lifespan. When a dicot seed, like this bean, is dropped in the ground and there is enough water and light, it may germinate. A dicot seed develops two cotyledons, or seed leaves, which are surrounded by a protective seed coat. The plumule is the embryonic shoot and consists of the stem and leaves above the cotyledons. The stem above the cotyledons is also called the epicotyl. The hypocotyl is the stem between the cotyledons and the radicle, or embryonic root. Germination begins when the radicle emerges from the seed. Next, the cotyledons emerge from the soil. A hook in the growing hypocotyl protects the plumule. After the hook breaks through the surface of the soil, the hypocotyl straightens out. This pulls the cotyledons and plumule into the air. The leaves of the plumule unfold and begin to perform photosynthesis. After their nutrients are used by the seedling, the withered cotyledons fall off the plant.
From these two videos, you see that several plant species which are used for further cultivation for specified purposes. Mostly, seeds are used for cultivation of this plant, but for many plants which are capable to grow by seeds and stem, cutting mode both root, stem, leaf, or their modified forms are also registered as propagules for development of the plants. Agritech is an important aspect for cultivation of the plants or crops. Cropping plants, varieties, and degrees of cultivation is variable based on different climatic conditions over the world. By lifespan of plant, we can say that perennial is a plant that lives for several years. Most perennials produce many times during their lifespan. An annual is a plant that completes its life cycle, grows, flowers, and produces fruits and seeds, and then dies within one growing season. So virtually all annuals are herbaceous plants. A biennial is a flowering plant that takes 2 growing seasons to complete its life cycle. So plants grow by producing new cells in a region of active cell division called meristems. Growth that increase the length or height of the plant is called primary growth, but growth that increase the width of stems and roots is called secondary growth. Let's see by the image and learn more about the plant structure.
A meristem is a growing region in a plant. Apical meristems are the growing regions at the tips of stems and roots in plants. Most plants grow in length through apical meristems. Some monocots have intercalary meristems. Intercalary meristems are located above the bases of leaves and stems and allow leaves of monocots like grass to regrow quickly. Gymnosperms and most dicots have lateral meristems, which increase the diameter of roots and stems.
So primary growth, what can we say about this condition? Apical meristems that are located at the tip of the stem and roots produce primary growth through cell division, of course by mitosis, right? The new cells produced by apical meristems differentiate into the primary dermal, ground, and vascular tissues of roots, stem, and leaves. These are the parts of plants. Some of the cells produced by the root apical meristem also become part of root cap. From this picture you can see how the shoot tips of callus plants, apical meristems, these are the cells under the microscope, and this is the root of the radish plant. Secondary growth is produced by cell division in 2 meristems, while primary with one, which form in cylinders near the outside of woody stems and roots. One meristem called cork cambium lies within the bark and produces cork cells. The other meristem called vascular cambium lies just under the bark and produces vascular tissues. The secondary growth, a woody stem, develops in 3 steps. First, a young woody stem has a ring of vascular bundles between cortex and the pith. The second, a vascular cambium forms between the xylem and phloem. In each vascular bundle, cork cambium forms under the epidermis. Third, in a mature stem, vascular cambium adds new layers of secondary xylem and phloem each year. The layers are called annual rings. You know it from 7th grade how we can count the annual rings and say the age of the plant.
A young stem is covered by epidermis and contains pith and cortex. A ring of vascular bundles containing primary xylem and primary phloem separate the cortex and the pith. Vascular cambium develops between the xylem and phloem in the vascular bundles. Secondary xylem is produced toward the inside of the stem, and secondary phloem is produced toward the outside of the stem. As the stem grows in diameter, the epidermis is stretched and broken. The cork cambium forms and produces a layer of cork around the outside of the stem. Eventually, the vascular bundles merge into solid cylinders. The cork cambium, and secondary phloem cylinders make up the bark. The vascular cambium and secondary xylem lie inside the bark. Each year, a new layer of secondary xylem is produced by the vascular cambium. In the spring, when water is plentiful, the xylem is made up of wide, thin-walled cells. This wood is called springwood. In the summer, when water is more limited, the xylem is made up of small, thicker-walled cells. This wood is called summerwood. Older portions of xylem eventually stop transporting water. These portions are called heartwood and often become darkened with organic compounds. The lighter, functional xylem is called sapwood.
So by plant development, we can say that it makes the new cells in meristems. And summarizing, these cells differentiate and replace or add to existing tissues, thus the plant continues to develop throughout its life. Many cells in the mature plants can activate all of their genes. These cells can undergo differentiation and develop into a mature plant. A technique called tissue culture is used to grow new plants from issue that can reserve its development. Finally, I want to conclude that required for take care of each stage of plant cultivation, soil quality, plant variety, climatic condition, cultivation technique, market value, and etc. are leading factors for high production and economic growth. Thank you for your attention.